So, yeah, as it says here, my co-authors are Louise and Abby, demonstrating that they've got good dentists. And as there was, you saw, I put a question in the question box because it turns out that pre-eclampsia and long COVID do actually share quite a number of similarities. Now, there's obviously no doubt about the infectious origins of long COVID, and the first part of my talk is going to be rehearsing the arguments that actually preeclampsia also has an infectious origin, and the second half is what we can do about it. Now, of course, the old word for preeclampsia was endotoxemia of pregnancy, and the belief that there was an endotoxin circulating in the blood, which experimentally you could easily demonstrate, did bad things. And although Anatine is going to talk about this later, and I won't dwell on it, we fully accept Chris Redman's wonderful two-stage model of preeclampsia, in which you will see I have underlined the word oxidative stress and hypoxia. And the question is, we agree that they're part of the features, but where do they come from? What causes them? And one of the things that is not news that is able to cause oxidative stress and inflammatory responses is, of course, bacterial lipopolysaccharides from gram negatives and the equivalent from gram positives. And there's a lot of them in human beings, and they're actually known as endotoxin. And there is a model of preeclampsia going back quite some years now, in which you induce it by giving a small amount of lipopolysaccharide. So, according to one view, the reason it's called endotoxemia of pregnancy is because it's actually caused by endotoxin. And we developed those ideas in much more detail in a couple of reviews, or Louise and I did, over 100 pages. I'm not going to be able to take you through all the evidence that led us to the view that there was a good case to be made that dormant microbes were responsible for the origins of preeclampsia. But the reason I have to go through this is because a paper in Nature appeared that said the exact opposite, which of course everyone will have read. And if they believe the headline title, I'm barking up the wrong tree. Clearly, I don't think I am. And the argument behind it was the lower paper from the same group that recognized that many of the reagents we have actually have microbes in them. And if you don't clean them thoroughly, your samples that you interrogate with things like polymerase chain reaction reagents, DNA polymerase and so forth, are likely to light up the microbes, but the microbes come from the reagents, not the samples. But unfortunately, they didn't follow their own advice, and they did not clean their reagents. They also completely ignored all the evidence that I'll rehearse some of shortly. And the reason I know they didn't, apart from the fact that they told us, is that in the extended data, they did a control experiment in which they purposely added some microbes, it was S. bongori, and asked the question what fraction of the reads were from the things that they'd added. And the answer was about half, which meant that the background was about half this number, which ended up being about 44,000 contaminants per gram. If that was in urine, it would be close to bacteriuria. It was a massive amount. And of course, it completely overwhelmed the natural microbiome that we're looking for, which is dormant. And not in that paper, in many other things that we'll look at, the clever contaminants only appear in the disease, not the control. So that's very smart of them. Many, many papers showing that there are, in fact, microbes in the womb and related reproductive tissues. And not just there, it is absolutely well known in biology that we are all dripping with dormant bacteria and viruses. Here's a paper from the tumors in Science, another one on tumors from Nature. Helicobacter is present in about half the population, mostly doesn't give us ulcers. Mycobacterium tuberculosis is present in about a third of the population, almost none of whom go on to get TB. It is not news that we are dripping with dormant microbes. Except for a clinical microbiologist, these things don't tend to exist, because the clinical microbiologists will tend to look at what's plated out, and if they can replicate, they're called viable, and if they're not capable of replication, they're not seen at all. And by definition, dormant bacteria do not replicate on a Petri plate unless you do something nice to them so that they're not dead, but you can get them back later, known as resuscitation. In clinical microbiology, such dormant bacteria tend to be called persisters, but it's the same principle. An organism that won't plate out when you put it on a Petri plate in which it should normally grow, and yet it is not dead because you can get it back to a state in which it will. So all of these microbes, and including in nature, if you're a microbe in nature and you stop growing because you've lost nutrients and signaling molecules, if you die, evolution sees you off. You do not die, you go dormant. Most bacteria in nature are dormant and have necessarily evolved to be able to do that. And how do you detect them if you can't count them on a plate? You look down a microscope or you look molecularly for their sequences. Plenty of papers looking down a microscope and find them. Here's one. They're all referenced in the review, so I'm going to belt through these really. And many, many examples in which people find far more microbes and the effects of microbes, such as immunoglobulins, in the blood and urine of individuals who've got pre-eclampsia. One of the sources is the oral microbiome, because people will tend to bleed in their teeth, and therefore it's a two-way street. The bugs get a direct route into the blood under those circumstances. There's an odds ratio there for 2 oral bacteria of up to 9-fold. These are big odds ratios. Also, if antibiotics, if the cause is microbial, antibiotics should have a big effect. The Gondii example is particularly nice because you have to take spiromycin for a year if you're infected with Gondii, and obviously that can cover an entire pregnancy. And so the people in this study, which is cited in the review, did this, and the odds ratio, the ones who were infected with Gondii that might have made you ill were actually protected by an odds ratio of 11-fold. This is pretty strong evidence. I have an easy explanation for it. Anyone else is welcome to their own, but there's a pretty obvious one as to what it is. It is PE has an infectious origin, and the presence of these antibiotics stops it. And finally, in the second review, we asked the question, where do the organisms mostly come from? And the possibly surprising answer, because everyone's forgotten about the bloke 9 months into the thing, probably the answer is the bloke. And so that's what the story is. So for the second part of the talk, I'm going to talk about the fact that oxidative stress can be caused by LPS shed by microbes, and the second half is what can we do about it. And ergothioneine, which is mostly in mushrooms, or the chief dietary source, is this molecule put up there. And it's a potent antioxidant, both on its own and by waking up a transcription factor called NRF2 that turns on a lot of so-called AREs, antioxidant response elements. We have evolved to have a transporter for ergothioneine, rather suggesting that it's important evolutionary. It may even be a vitamin. And in the paper cited at the bottom, we predicted that actually it could be a good therapeutic or preventive for preeclampsia, given that the poor placentation part, that ship has sailed. We can't do anything about it. The oxidative stress part, we can hope to do something about it, because that's the second half of the 2-stage model of preeclampsia. And we wrote a big review on this. You can find it in the references at the end, and on my website where I put it. I'll put a link. And not only here, these people asked the question. They looked at the relationship between the metabolome of 3,000 Swedes, this is Lund University, and the molecules that were most correlated with the people not getting a heart attack, so pure cardiovascular, in the next 3 years. And you can see ergothioneine won by a mile. The second is proline betaine, which is closely related and comes from citrus juice. And so we did these experiments with Cahal and others, and indeed showed, or they showed, that in the RUP model, which is considered an okay model, we recognize that none of the models are terribly good for mammalian biology, but the ergothioneine in their drinking water did prove protective of, as judged by the weight of the pups after they'd been born. And now, when I first met Louise way back when in Manchester, we started doing metabolomics together, and that proved very successful. And more recently, we've reverted to reanalyze some of the scope samples, and this audience will know what scope is, a very big trial of an analysis of pre-eclamptics and non-pre-eclamptics. They didn't have any dietary interventions or analysis, but we used a very good quantitative metabolomics method to ask the question, what is the relationship between the amount of ergothioneine in their plasma in the scope samples versus whether or not they got preterm, term, or no pre-eclampsia? And that was published relatively recently, and the reference is there. And so if you look at the whole distribution of ergothioneine in the population studied, which was 400 and something, you can see that actually it's not really a normal distribution. It's got a long tail, and we'll come back to that because it's important. There was no relationship between ergothioneine and BMI, and there was a weak one with maternal age, but in the positive direction. Obviously, maternal age tends to be more of a predictor of PE when it's going positive, but we're going to see obviously the opposite. So then we ask the question, if we look at the median levels of ergothioneine in the three populations, is there a difference? In other words, would I use this as a biomarker? And the answer clearly is no. As judged by the medians, ergothioneine is no different between no preeclampsia, preterm, or term. But if you ask a different question, so this is the classical frequentist statistics beloved of people wrongly, and we want like the Bayesian or machine learning approach that asks a direct question. If I take the 90th percentile and ask the question, what is the relationship between whether or not our people got preeclampsia or not, if their ergothioneine was above the 90th percentile, what I find, same data just plotted in a different way, is that only one person out of the huge cohort got term preeclampsia, no preterms. And so the odds ratio, if your ergothioneine level is above that point, which is the 90th percentile, chosen purposely, it's not arbitrarily. I mean, that is a common way of choosing for these kinds of analyses. The odds ratio is massively in the favor of not getting preeclampsia. So bottom line, frequentist stats showed there was no difference. Machine learning shows there's a big difference, provided, and the provided is that you're above a certain level. Obviously, this is at least highly suggestive that ergothioneine would be protective if it was given as opposed to just randomly measured in the populations of interest. So, a swift canter, but we're nearly there. Dormancy is actually normal for microbes in their natural environment. Clinical microbiologists don't tend to recognize this, but that doesn't make it untrue. Of course, the poster child for microbes causing supposedly non-infectious diseases is Helicobacter. We actually think, and you can read the reviews, that all chronic inflammatory diseases have a infectious origin, whether bacterial or viral, and there is a ton load of evidence if you care to read our reviews. I also didn't discuss iron dysregulation, which is part of the story because there wasn't enough time for that. But we think that there's very strong evidence suggesting that testing ergothioneine, or if you prefer mushrooms, would be a good preventive of preeclampsia via the antioxidant effects that it's demonstrably capable of, and we are trying to get an RCT funded. So hopefully, if anyone ends up refereeing it, you will look kindly upon it, because that's the evidence I've given you today. All of the publications are on my website, and they're all open access, so they can be nicked and studied. And we also wrote this book trying to understand why people believe bonkers things despite all the evidence. And so with that, I thank you for your kind attention, and I have done exactly 15 minutes.
Okay, thank you, Doug. That's very interesting and equally confusing with it, as a lot of your presentations are. I think that you bring in so much information from it, and it's you know, it conjures up questions that your graph showing the 90th percentile work. It does suggest, though, that if its levels are normal, that it's not predictive. So this is not this is not absolute, or is it relative risk that people will have? I mean, this inflammatory source of preeclampsia, is that true in everybody, or is it true? Is it also true in normal pregnancy? So therefore, it doesn't differentiate or what?
So of course, everybody is different. If I pick 10 genes out of the 25 and say there's a variation of their expression level of twofold, I can explain 2 to the power 10, which is 1,000-fold. So with 10 genes, I can explain anything, or 10 variables. From that point of view, what's surprising is not that people are different, but that they're similar. I think the argument is that yes, there is a variation. Some people are much more sensitive than others to the getting preeclampsia, mostly for lifestyle reasons, some genetic reasons, but very marginal. It's pretty well entirely lifestyle. And others are relatively resistant. And we don't know the causes, otherwise we wouldn't be having these symposia. But what is very clear is that if you managed to get above that magic 90th percentile, you can effectively overwhelm all the bad chances and risk factors that would otherwise increase your likelihood of getting it.
So I mean, we're actually a minute or two ahead of time, but so is what you're finding, is this something which can be, if it's overwhelming, can be a cause of forming the disease, or if it's less active, then it could be a mediator who aggravates other inflammatory processes which may or may not come from the center?
Correct. I mean, essentially, it's this balance, and people often draw seesaw diagrams. We've done it in one of our reviews, in which there are some things that pertain towards getting PE or whatever the disease of interest is, and other things that make it less likely that you will, both maternal and other factors. So yes, it's a question of upping the ante more and more in your favor. For some people, you probably don't need hardly any. But other people, you need loads and loads. We don't know which is which, so you might as well give loads and loads, because there's actually been studies. People have eaten industrial quantities of this stuff. It's totally safe.